So where do antibiotics come from? Well, antibiotics are naturally produced by some bacteria and some fungi, which is interesting. Antibiotics, again, are naturally occurring molecules. We humans use them to our own benefit, but it's not originally what they were produced to do. So these genera here, Streptomyces, Bacillus, Penicillium, and Cephalosporin, that was a tough one to say, all produce antibiotics naturally. Not every single species from these different genera produce antibiotics, but some species from these groups of bacteria do. So the first antibiotic that was discovered was penicillin, and that is a drug that we still use sometimes today. And this drug was discovered by biologist Alexander Fleming, and this was an accident. So basically there was contamination of a petri dish. So I don't think that we have seen this in any of our labs in particular, but if you take a look at what he's pointing at here, this is a fungal colony which is growing on a bacterial petri dish. And this, I doubt that this is the original colony or dish that he discovered penicillin on, but anyway, basically he observed that there was fungal growth on the dish, and this was a dish where they were also growing a bacterial culture, and he noticed that the bacterial culture couldn't grow adjacent to where the fungal colony was growing. So you see here, this white spot would be something like the fungal colony or the penicillium, and then this here is the bacterial growth, and in between you could see that there is a space between the bacterial growth and the fungal growth where there's no bacterial growth. That's because this area here that I'm coloring in is preventing bacterial growth because the fungal colony is releasing antibiotics, and the highest concentration is right here next to the fungal colony, and so we don't get any bacterial growth in that area. So what Alexander Fleming noticed was this lack of growth between the fungus and the bacteria, and that's when they came to find penicillin. So penicillin is produced by fungal species, in particular the Penicillium mold, and we still use penicillin today, although we know that some people are allergic to it. They have an allergic reaction, so don't confuse that with penicillin affecting human cells the way it affects bacterial cells. It's completely different circumstances causing humans to be allergic to penicillin. So how does penicillin work? Well, penicillin targets the cell wall, as we saw in this image here. And so basically, if a bacterial cell is exposed to penicillin, penicillin is going to bind to an enzyme which is in charge of building the cell wall. So if the bacteria is just kind of sitting there and not doing anything, then that's not a problem. But on the other hand, I'll draw this bacterium here, and it's not doing anything, that's fine. If on the other hand the bacterium is going to grow, then it needs to make more cell wall. And as you've seen in previous lectures, the cell wall of bacteria is kind of like a chain link fence in some ways. It's interconnected between multiple layers, and so in order to make a larger cell, you need more cell wall, and in order to make more cell wall, what you're going to have to do is get rid of some of the connections there, and then move these two pieces apart, and then add in new cell wall, right?